So what the aim is for today is to actually go through some of the important question words that come up in the exams, and then we're going to look through a past paper question to see how these question words are actually used in the questions themselves. So just want to see if I can pop. I can. Okay, brilliant. Okay, so we should be good now. Hold on, I'm just rearranging my screen so I can see everything I need to see, so I can keep an eye on you guys while I'm teaching. Right, now we've lost the start. Investigating question words, there we go. Okay, so in your syllabus, there's actually a whole page of question words, and they give you a sort of technical definition of what is meant by each of those question words. So I've just pulled out a few, because these are the ones that sort of come up really often and the ones that people often get confused with. So starting at the very top, define. Okay, they could ask you to define something, give the definition of, define what is meant by. Okay, this is your standard definition. It's things like, what is photosynthesis, you know, define what a protein is. It's not something that's complicated. Define a tissue, you know, a group of cells that are all similar cells that are working together to perform a single function. Okay, so those standard definitions that you find in your textbook and you find in your notes, learn them, because then when you come to it, it's a case of just regurgitating that definition. Okay. Okay. Sorry, I'm just playing with Zoom as well in the background. Okay, so next one, state. In this case, you're asked to state something. State what is. State the type of. Okay, and here you're just looking for a short, concise answer. State where this is found. State what the function is of that. We don't want to explain, we don't want to describe, we just want to say the function is XYZ. Okay, if they ask us to explain, maybe we're going to change color here because these next two are ones that often get confused. Explain and describe. So to explain something is to give the biological factual reasons behind something. Okay, so it's why does it do what it does? How does this happen? What's actually happening behind this general statement? Okay, to describe something is just to give the main points in words. So for example, you could be describing something that's from a graph or a table, or you could be describing a process. X, Y, Z, 1, 2, 3 is my process. Okay, if you're describing from a graph, it goes up, it stays the same, it goes down. That's the sort of thing we're looking for. Okay, so if you're describing, you're just saying, I have a lunchbox. If you're explaining, you're saying, I get hungry, therefore I bring lunch to work so that I have something to eat during the day. Okay, so there's a big difference between describe and explain. And it's really important that you take the time when you're reading the questions to actually go and look, what is my question word? Is this question saying describe? Is this question saying explain? Is this question saying define? You could be asked to combine them. You could be asked to describe and explain. So describe what the graph looks like and explain the reasons for it. So it goes up because X, Y, Z. It stays the same because X, Y, Z. It goes down because X, Y, Z. Okay, so you're getting the idea here that we're describing things, we're just saying what it is. We're just giving those main points in words. Whereas if we're explaining, we're giving the because, the why behind it. Okay. Another one that can trip people up because it's unusual and unexpected sometimes, suggest. Okay, suggest means this is something you haven't studied. This is something that's not in your textbook, but it links into things that you have studied. It links into things that you have learned. And that's why I'm always saying that it's so important that you learn all the facts, that you learn all the definitions. You have to have that knowledge in your head. It needs to be stuck there. Because the thing is, when it comes to suggest, you're going to be faced with a novel situation, something you've never seen before. But you're going to use your knowledge. You know A, B, and C. Therefore, if you know them well enough, you can pull on them and you can say, okay, I'm looking at this. A, B, and C is going to help me. Therefore, I can suggest something. So you're not just suggesting some random sort of answer. You're thinking about what is it that I know and how is this going to help me? Okay, so your next round of words that sort of sit together are calculate, measure, and determine. So here we're getting slightly more logical, slightly more mathematical. Calculate. We're going to do a calculation. Yes, you get a calculator when you do your biology exams, but it's really important that you remember to show your calculations. Okay. If you are asked to measure something, you're going to be using some sort of instrument. As you're not doing a prac exam, the most likely instrument you're going to use is a ruler. So please make sure you can measure with a ruler properly. And then if you're going to determine something, sometimes it means calculate, sometimes it means use a graph. So you're going to have to understand that word in the context of your question. Okay, so calculate, measure, determine. We're looking for a little bit more mathematics here. Okay, so I see we've got a whole bundle more people. So that is the end of that section. So I'm just going to quickly grab a register of everybody who's in now. Just to make sure that I am on top of everything. Then we shall go back to our lesson. Right. So it's all very well learning all these random terms and types of questions. State, explain, describe, suggest, calculate, measure, determine. And there's a few other you'll come across, but they're not so common. Okay, so these are the ones we're going to focus on today. And what I've done is I have pulled a past paper question that we can actually see these things and work through them. So what we've got is Summer 18, Paper 41, Question 4. So just a little note, Summer 18, that means, whoops, May, June. Seems like my spelling has got out the window today. May, June, because it's the Cambridge summer. Paper 41 is paper 4, because we were doing the extended course here. And paper 1 is the variant. So 41, 42, and 43 are all different versions of paper 4. And then question 4, pretty sure you know what that means. Right, so this is looking at some of the work that's further on in the course. So it's not a case where I'm expecting those of you who have sorted to be familiar with what we're looking at now. But it doesn't stop us having a good look at the questions themselves. Okay, so we have the blood flow through the skin can be investigated using a flow meter. Okay, figure 4.1 shows a flow meter above a section through the skin. So at the top here we have our flow meter that I presume is measured to connected to some sort of measuring device. And then we have our skin, and we're reminded this is not drawn to scale. Okay, so we have different layers of skin. If you have studied the skin already, this should be looking familiar to you and should be something that you can label now. Okay, we're going to work through things gradually. Okay, first thing, this one's going to be an artery. This is I'm not sure if you can see my pen, so let me label. Artery and a vein, okay, because we can see the blood flow coming in. Okay, remember we go from an artery into an arteriole, into capillaries. Then the capillaries join together to form venules before we get back to the vein. And then this here is a shunt vessel. Okay, so what looks like a squiggly thing with roots or trees, this is a nerve cell. And we can see here we've got lots of connections with the skin. So we have here a sensory neuron as opposed to a motor neuron. Okay, so we've got an idea of this. We're pretty sure now what we're looking at. And we've done this before we've even looked at the question. Because remember, whenever there's information given in the question, that's information that's useful, it's helpful, it's going to be used in the question. So we want to understand that information properly, exactly the same as in the prac paper. Right, so getting on to the questions, look at those 3 question words. Those 3 question words are all state. State, state, state. So state the name, state the type, state the name. So we're looking for really concise answers. Just the name, the types. So the name of cell P, we already know that one. This is a sensory neuron. State the types of blood vessels labeled Q, S, and T. Right, so where'd Q go? Q is there, he's coming back down, we've already named him. He's a venule. S is going up to the capillaries, so we have an arteriole. And T is our capillaries themselves. Okay, so venule, arteriole, and capillary. State the name of the tissue at R that provides insulation. So you've got 2 clues here. One clue is the location. The other clue, they've given you the function. So here's the location of R. It's this layer here. Providing insulation, well, that has to be our fat tissue. And again, just the name, we're just stating here. So moving on to the next question. Oh my word. Now we get a whole lot of information. So make sure we read it. So what's happening? Blood flow through the skin of some volunteers was measured with a flow meter when their skin was exposed to different temperatures. Okay, this is making sense. Okay, because we know that blood flow to the skin is going to change at different temperatures, so we can measure it. That's cool. Right, capsaicin is a compound that gives people the sensation of feeling hot when it is put on the skin. Researchers applied capsaicin to the skin of the volunteers and again measured the blood flow through their skin at different temperatures. This is the stuff that's in chili peppers. This is what makes your mouth burn when you eat chilies. Okay, so what they're saying is we have this fake feeling of heat. So how is that going to affect the blood flow? Is the blood flow going to respond to the fake heat as well as the real heat? How does that two interact? Okay, so figure 4.2 shows the results. Here's a graph, but we want to now dissect it. Okay, so what is our dependent variable? What is our independent variable? Remember, independent always sits on the bottom. So independent is temperature of the skin surface in degrees C. So what temperature did we expose that skin to, right? Dependent is average blood flow as a percentage of maximum blood flow. So you could have no blood flow, 0%, all the way up to the maximum blood flow of 100%. And this is what we're measuring. And it's dependent because blood flow depends on skin temperature. Now let's look what we got. We've got two lines on our graph, sorry. And they're labeled clearly without and with capsaicin. Okay, so we can see that they start out the same, but then with capsaicin has a steeper curve going up and our blood flow is increasing more than the without capsaicin. So, what's the actual question that we're dealing with? First question says, use the information in figure 4.2 to describe the effect of increasing the temperature of the skin surface on blood flow to the skin without capsaicin. So, I spoke funny for a few words. Right, information in figure 4.2, describe without capsaicin. Okay, so we are only dealing with this curve here. We're not bothered by the other curve, we are just describing one curve. Okay, so It stays the same, it goes up. That's our general pattern that we're describing. But we want to get marks. So remember they say use the information. That means we need to pull some numbers off here. So it stays the same at what percentage? It goes up to what percentage? So if we pull this together into an answer, we can say from the beginning, I've scribbled on it a bit now, but I hope you can see these first three are the same. So if we read our scale here, we've got 5 blockies to 10, which means it's 2, 4, 6, 8, 10. So these are at 4. So we can start out by saying that the blood flow is constant at 4%. Up to, where does it go up? Well, the first record we have of it going up is here at 25 degrees C. Okay, so we've given a description of the shape of the graph and we've attached data to it. From 25 degrees C, there is a slight increase in blood flow. Okay, so that slight increase goes, we could say maybe to about here somewhere, and then it starts getting steeper. Okay, so a slight increase in blood flow. Okay, the increase then becomes steeper. To a maximum blood flow of 100% at what temperature? So if we look at it, we've got 40 up to 5, we've got 10 blockies, so we're going 41, 42, 43, 44, 45. So if we bring up, it's easier to do this on a paper than a screen. That was our 40, so this one is 41 degrees C. So for maximum blood flow, 100% at 41 degrees C. So we've described our curve. We had constant, we had up. What was our constant? Where did it change? What was our maximum? So each time you're looking at a graph, you're looking for things like that. You're looking for, just add in, you're looking for up zones, increasing zones. You're looking for constant. You're looking for fluctuating. You're looking for decreasing. We should be technically correct. Let me just change that first one. This would be an increasing, not an up zone. Okay, so increasing. Helps if you can spell increasing correctly. Increasing, constant, fluctuating, decreasing. These are the zones that you're looking for on your graph. Okay, then you're also looking for data for details of edges of zones, and you're looking for your maximums and your minimums. Those are always interesting. Okay, so you have a look at your graph, you understand what your graph is showing you, and then you dissect it. Next thing. Now we have a different type of question. Explain the mechanism that increases blood flow through the skin. So let's get back to that red pen. Because we have an explain, we have a state, but this state is accompanied by a space for working. Then we're going to have another explain, Then we're going to have another explain. Then we're going to have another explain. Okay, but there's a slight differences between all these explains. So, just watching the time a little bit because we're going to have to not go into quite as much detail for these next questions so that we can get through them and we can understand all the different explains. Okay, because the aim of this session is looking at the question words rather than the actual content and the knowledge and understanding surrounding temperature control. Okay, so if we ask to explain the mechanism, we want to know the biological factual details of how blood flow increases through the skin. What happens? How is it initiated? How is it controlled? How is it brought about? What happens? Okay, so your sensory neuron detects the temperature, your hypothalamus is monitoring your core temperature, we're sending signals to through the motor neurons to the effectors, and then we have all these different muscular contractions in the arterioles and the shunt vessels and relaxations and blood flow changes. Okay, so that's what we're looking for here. But we want, and I say it again, the biological specific facts. We need to be detailed for an explained question. Okay, next one says, state the difference between average blood flow for treatments with and without capsaicin at 35 degrees C. Now, a state question means just give the answer, but they've also given you here space for working. So, one thing to look out for is your mark allocation. Okay, because your mark allocation gives you an idea of how much should I be doing. If they're saying state, they're saying you can just write down the answer. 1 mark makes sense. That's a state. If you're not quite so confident, they've given you that space for your working. So you can go to your graph and you can read off your graph and you will get that without is 20%, with equals 66%. So your difference is 66 minus 20, which is 46. So 46% is our difference. Now the thing is here that we didn't have to do the working. Other questions, you do need to show your working. So it's better to be in the habit when they give you space for working, build that habit of putting working in. Okay, then you're covered, you know you're safe. Right, so the researchers thought that capsaicin stimulated receptors in the skin. Explain the process by which capsaicin could reach these receptors. So here, in this first explain, we were explaining a mechanism. In this second explain, we are explaining a process. Okay, so here we need to think, what is the process? Okay, the process is diffusion. See it coming back. We're going to take all the different levels, early in the course, late in the course, mix them all together in one past paper question. Okay, so once you've established that your process is diffusion, then how does the diffusion happen? Give me more explanation. Don't just state the process. We want to explain it. So we need more detail. Okay, diffusion is moving from a high concentration to a low concentration. We've got to get across the epidermis, got to get between epidermal cells, we've got to get into those sensory neurons somehow. Okay, so there's all these sorts of things that you've got to consider when you're explaining. You need that detail. Right. Now we have another different explain. This one says, explain the importance. Haha. Now, we don't study importance as such. Okay, we study biological facts. So when you're explaining an importance, you are now looking at application. So here we are applying everything we have ever learned about the importance of temperature in humans. So we're pulling from all sorts of different places. So we're thinking about things like enzymes. We're thinking about optimum temperature for enzyme reactions. We're thinking about things like active transport. Active transport occurs through a cell membrane, and it relies on carrier proteins. Remember, proteins are temperature sensitive. So, yes, we've got to maintain this temperature for our enzymes. We've got to maintain it for any other kind of protein as well. Okay, so you've got to take it to that little step further, not just straightforward steps, not just straightforward processes here, not a straightforward mechanism, but that application, okay? We can take it to a broader scale as well. Hyperthermia and hypothermia. We need to avoid those. We need homeostasis of the body. Okay, so all of these sorts of things would come into one of these questions. We're explaining an importance. The last one, luckily I've got a minute and a half to go to get through this one, because this one is an important one. Because this one says, explain how coordination by hormones differs from coordination by nerves. This question is about hormones. This question is not about nerves. Your answer is about hormones. Your answer is not about nerves. Okay, because we are explaining coordination of hormones. Coordination by hormones. Specifically, what is different to nervous control? So the fact that hormones are involved in control, we don't care, because nerves are also involved in control. But it's about how those hormones are different, how their job is done differently. That's what we're looking for. So when you got a question like this and you ask, well, why does this differ from that? Remember, you got to focus on, what are they asking me about? Okay? Yes, I want the aspects of it that are different from that, but what is my core question about?